Thank you, Andreas. So I'm very pleased to have this opportunity to join in this discussion of who are we. And I'd like to do this by considering two things that you've already heard about. Things that I myself have been studying for more than 40 years. Our genes and our brains. In the next 20 minutes, I hope to convince you of the value of thinking about who we are by focusing on our genes and our brains. First, our genes. As you know, every cell of our body has chromosomes that contain genes made of strings of DNA. These strings are made of 4 building blocks called bases, and these bases are symbolized by the letters A, T, C, and G. Each of us gets 3 billion of these building blocks from each of our parents, so that we have 2 complete sets. And each set is arranged in a seemingly boring sequence of A's, T's, C's, and G's, like that shown on the slide. But we've learned to read and decipher the meaning of the sequence, and we've learned as a result that it's far from boring. In fact, this sequence, those A's, T's, G's, and C's strung together in that specific sequence, has a lot to tell us about who we are. So, what does this sequence mean? It was discovered in the 1960s in experiments that I played a very small part in, that these sequences are translated into the language of proteins. Unlike DNA, which has only 4 building blocks, A, T, C, and G, proteins have 20 building blocks called amino acids. And each of them is represented by a specific sequence of 3 bases. So each sequence of 3 bases is a code, a genetic code, that tells the cell to insert a particular amino acid into a particular position in a particular protein. For example, on the slide you see 3 bases, A, T, G, that are translated into one type of amino acid that's symbolized by that little yellow ball, while the next 3 bases, C, T, A, are translated into a different amino acid, symbolized by that little pink ball. And each of them is hooked together to make a protein. Once the protein is made, its job is determined by the sequence of the amino acids, the yellow and pink and purple and what other kind of balls it has. And those are determined by the sequence of the DNA. So what do these different proteins do? Some proteins are structural materials like the collagen in our skin. Some are enzymes that digest things that we eat and turn them into other key chemicals like hormones or neurotransmitters. And some of these proteins control the function of other proteins and of particular genes. So that in the end, all of us are molecular networks of proteins and genes and the molecules they make that keep talking to each other and that make us who we are. But it's not that simple, and it's much more interesting. Although each of us has 20,000 human genes, they are not exactly the same. Instead, there are different forms of each gene, and each of us, except for identical twins, inherits a different combination of variations. Furthermore, even tiny variations that change a single amino acid may change the properties of the whole gene, of the whole protein. It's just like changing a single link in a steel chain by making that link out of a string or a piece of plastic or a piece of rubber. That change in the properties of the single link in that steel chain can have a very big effect on the function of the whole chain, on how easily it bends or folds or breaks. To show you the significance of inheriting different forms of a gene, I'd like to tell you about two examples. The first is called apolipoprotein E, or ApoE, and it has 3 common forms. The second protein is the serotonin transporter protein, or SERT, and it has 2 common forms. And as you will see, each form influences who we are. So here are the 3 common variations of the ApoE protein. And the variations are all found in positions 112 and 158 of the chain of 299 amino acids, which is the size of this particular protein. The commonest form of ApoE is called ApoE3, and it has an amino acid called cysteine at position 112 and an amino acid called arginine at position 158. The rarest form, ApoE2, has 2 cysteines, one at 112 and one at 158, while ApoE4 has 2 arginines, one at 112 and one at 158. I've also listed on the slide the frequency of the different forms in a group of people, like the group of people in this room. So most of us have 2 copies of form E3. We are E3. On the slide, 55% of people in the population that was studied are E3. But about 2 in 100 of us have 2 copies of E2, which, as you will see, is of E2, which, as you will see, is the most desirable form. And 1 out of the 100 of us have 2 copies of E2, which is the most desirable form. That is, E4 is the least desirable form, and E4 is the most desirable form. And others amongst us in this room have various mixtures of E2, E3, and E4. Now, the variations of the other gene I told you about, the SERT gene, are of a very different kind. In this case, the variations are found in the part of the DNA called the promoter, the part of the gene that controls the amount of the protein that is made, while the structure of the SERT protein is exactly the same in both gene forms. So the long form of the gene makes more SERT, while the short form makes less SERT. And about 57% of the genes in a typical group of people, like the people in this room, are the long form. So the lesson from this detailed study of these and also the many other genes the whole 20,000 genes that are the human genes, is that all of us are both the same because we all have those 20,000 human genes. But also, all of us are different because each and every one of us has particular combinations of the many different variations that make us ultimately genetically unique. The other thing I'd like to leave you with in this part of the talk about genes is that it's becoming possible for each of us to get a complete and total sequence of our personal DNA. Everybody in the room can now get this, or will soon be able to get this. In fact, a company called Complete Genomics has just announced that in the next few years, it will be doing this for $5,000 per person. This is an enormous amount of work. $5,000 a person. They will give you, if you wish, your complete genome sequence, base by base. Furthermore, they and others hope that this sequence will eventually become available for $1,000 or even less. And once this is done, people can, once they get their complete genome base by base, the entire sequence, they can do with it what they wish. They can use it for their own personal purposes, or they can also share it by combining the information from their personal genome with the information from all the others. And in fact, Steven Pinker, the last speaker in this session, is one of a group of people who are now planning to not only have their DNA sequenced, but also to publish it in whole or in part on the internet so that that information can be combined with the information from many other people for a variety of hopefully useful purposes. Now, in case you are impatient and want immediate information about your genome, there are also cheaper but less detailed ways of discussing, that is, of obtaining this information that are already available. In fact, in the last issue of Time magazine, which the magazine calls the invention of the year, there is the description of a much more limited analysis of a human genome by a company called 23andMe, and this can be ordered by anybody on the internet for $399. It doesn't give you a complete genome sequence, but it does tell you a great deal about your specific set of genes. But before you rush out to buy this service, please be aware that we are still a long way from figuring out to do with this type of information. The technology for genotyping people has advanced incredibly quickly, but interpreting it and figuring out how to apply it remains rather challenging, and the dangers for misinterpretation remain quite considerable. Now, the second part of the who are we that I'm going to discuss is our brains. And so now that I've explained a little bit about our genetic differences, I'd like to turn to a consideration of those. Brains are, of course, very complicated structures that are built of circuits. You've already heard a little bit about this. These circuits are connections of brain cells that together control particular ways that we think and feel and behave. For example, the diagram shows components of an important brain circuit that contains or that controls some of our emotional reactions. The components are collections of specialized nerve cells with names like the VTA, the septum, the nucleus accumbens, the amygdala, and the prefrontal cortex. And these parts are hooked together in all of us. But the strengths of particular connections varies from one person to another. And this controls certain features of who we are, such as our tendency to feel happy or our tendency to feel sad. So what tells an individual brain to build up these circuits and put them in the way that installs our own personal settings? It turns out that the main contribution of this process is our genes, but they don't all work at once. In fact, it takes about 20 years to build up each of our distinctive brains. And this sequence of processes is controlled by a series of internal clocks, like the one which turns on the gigantic brain changes that occur at puberty. During this 20-year period, genes build certain brain structures, try them out to see how they work, and then modify them. By responding to internal signals and feedback from our own personal experiences, the genes, in concert with the feedback from our environment and our personal experience, gradually sculpt our personal and unique brains. Now, not all circuits in the brain are spread out all over it in the long distance that I've showed in the previous slide. Some involve nerve cells that are packed closely together in the outer layers called the cerebral cortex. And once the genes that influence these tiny circuits act, and one of them is our old friend ApoE, important changes result. And one way we know about this is by measuring the thickness of the cortex in teenagers with an MRI machine like the one used to make routine medical diagnoses. It turns out that teens with an E2 variant have the thickest cortex. Those with copies of E3 have an average cortex, and those copies with at least one copy of E4 have the thinnest cortex. Furthermore, the thicker the cortex, the better their memory for certain kinds of tasks. But the effects of the ApoE genes aren't over in our teens. In fact, genes keep doing things and doing new things in the course of our lives. So later in life, they have another big effect. They influence our risk of developing Alzheimer's disease. In this diagram, the risk of developing the disease is symbolized by the size of the figures of the people. The bigger the figure, the greater the risk. The majority of us who have 2 copies of E3, shown in gray, have an average risk. Those with 2 copies of E4, shown in blue, have the highest risk, while those lucky people with 2 copies of E2, shown in green, have the lowest risk. The 2 forms of SERT, that other gene that I told you about, also have complicated effects. These proteins influence circuits that use a substance called serotonin, and some of these circuits influence our tendency to become frightened, and this can be measured by examining people's brain activity. One way to do this is to show them pictures of frightened or sad faces to see how their brains react by measuring the brain responses in an MRI machine. The machine measures the activity of the parts of the emotional brain circuits, such as the amygdala, and the intensity of the reaction is shown in a color, with yellow being the most intense. So in the figure, that bright yellow spot in the brain is the very active right amygdala in a person with short SERT. And that is not an isolated finding. The bar graph shows that a group of people with short SERTs have a stronger response a more intense fear reaction than those people with long certs. And this fits with other types of studies that show that people with short certs are more likely to get frightened and depressed. To get back to the opening question of the session, who are we? One big answer is that all of us are both the same because we share the same genes and the same brain circuits, but we are also unique because each of us has our own unique set of gene variants and brain circuits. So just as our faces are similar but unique, so are our genes and our brains. So why should you care about all this? One reason should be obvious. Working out the details of what is shared and what is different is helping us learn ways to prevent and treat common human problems, such as Alzheimer's disease, anxiety, and depression. But there is also another implication of this knowledge, and I would like you to think about it to see if it also holds true for you. I personally find that understanding these building blocks of our nature increases my compassion for people, compassion for others, and when I'm in a suitably enlightened mood, compassion even for myself. Thank you very much.